Welcome to the first chapter. It is transport in plants. Plants, just like all other living organisms, do carry out life processes of growth, reproduction, metabolism, repair, and so many others. So they need a supply of essential materials to be able to carry out these life processes. Plants are autotrophs, which means they make their own food from simple molecules. This contrasts with animals which feed on ready-made food. Animals are heterotrophs. Plants manufacture food through the process of photosynthesis, mainly in the leaves. To do this and many other processes, plants require water and minerals which are absorbed from the soil by roots and transported up to the leaves. In the leaves, food is manufactured from carbon dioxide and water in the presence of sunlight. Once the food is manufactured, it has to be transported from the leaves to the storage structures, usually the stems and the roots. Now, all this means that a transport system is essential in the plant. In multicellular plants, most cells are way far from the medium and diffusion cannot adequately meet their needs. An elaborate transport system is thus needed to accomplish this. Materials are absorbed by the roots and transported via structures called xylem up to the leaves. The food is manufactured and is transported from the leaves via structures called phloem. Now, the xylem and phloem make up what we call vascular bundles. So we are going to look at each part of the plant, that is the root, the stem, and the leaves, and see the vascular bundles in them and how these structures carry out various processes. And we start with the root. The root of a plant anchors the plant in the soil, and the outermost layer of a root is called the piliferous layer. The apex of the root is covered by a root cap which protects the growing part of the root. As the root elongates, pushing through the soil particles, the root cap cells are shed off. The lost cells are replaced while the cells behind the apex are protected. Roots of aquatic plants lack the root cap. Why? Because they grow suspended in water and do not require protection. Internally, a transverse section of the root reveals the following layers of tissues, which include the epidermis, cortex, endodermis, pericycle, xylem, and the phloem. The epidermis is the outermost layer of cells that protect the inner tissues. The protoderm is the tissue at the apex that subsequently gives rise to the epidermis. The epidermis is made up of a thin layer of cells that are arranged over the entire surface of the root. The layer is 1 cell thick, and the thinness of the layer reduces the distance over which water and mineral salts pass. Some of the epidermal cells are modified to form root hairs. The cortex. The cortex is the tissue found between the epidermis and the endodermis. The cells of the cortex are of various types, which include the parenchyma and collenchyma cells. Collenchyma cells are angular and have thick cell walls, and these cell walls are thickened especially at the edges. The thickening of the cells is due to deposits of lignin on the cellulose cell walls. This thickening of collenchyma cells provides support to the root. The cells may have chloroplasts which carry out photosynthesis. This is a more common occurrence in stems. Such photosynthetic collenchyma cells are termed as chlorenchyma. Parenchyma cells are spherical in shape with thin cell walls. These cells can be squeezed into irregular shape. Parenchyma cells fill up the spaces between tissues. Where these cells have chloroplasts, they carry out photosynthesis. The endodermis is a single layer of cells between the pericycle and the cortex. It is made up of rectangular shaped cells that have starch and many mitochondria. Some of the endodermal cell walls are thickened with lignin. Root pressure is thought to develop within the endodermis. The energy to sustain the pressure is obtained from the hydrolyzed starch deposits, and the presence of many mitochondria indicates that activities of the endodermis have high energy requirement. The pericycle is a layer of cells found between the endodermis and the vascular bundles, and the pericycle gives rise to lateral roots. Xylem. The xylem comprises of vessels and tracheids elements. The xylem transport water and mineral salts from the roots to the leaves. We shall come back and look at the structure and function of xylem. Phloem, also part of the vascular bundle. The phloem comprises of sieve tubes, elements, and companion cells. Manufactured food substances are transported through the phloem. Companion cells have many mitochondria. Energy released by mitochondria is used by the sieve tubes to translocate the manufactured food. Now, the endodermis forms a continuous layer on the root. The cells of the endodermis have starch granules that provide energy for the cell's activities. The cell walls are made up of lignin, which is waterproof, and the lignin deposit form a band known as the Casparian strip. The cytoplasm is attached to the Casparian strip and water molecules pass through the cytoplasm. Pressure is generated in the cell to force water into the xylem. So we now turn and look at the main function of the root, which is absorption. And absorption is done by the root hairs, which are modified outgrowths of the epidermal cells. The root hairs are numerous, long and slender, and this provides a large surface area through which absorption of water and mineral salts take place. They are found near the root apex, and those that are located further from the root apex often peel off. The part of the epidermis where the root hairs grow is called the piliferous layer. So exactly how does absorption of water take place in the root hair? The root hair is of course surrounded by water and mineral salts trapped in between the soil particles. Water is absorbed by the root hair and passes all the tissue layers until it reaches the xylem by osmosis. How? When the concentration of water molecules is higher in the soil than in the cell sap vacuole of the root hair cell, water moves into the root hair cell by osmosis. This water dilutes the content of the cell sap vacuole of the root hair cell. Now, at the same time, the cells of the cortex have less water molecules than the root hair cells, and water moves from the root hair cells into the cells of the cortex by osmosis. The content of the cells of the cortex become more dilute than in the cells of the endodermis. Again, water molecules move into the cells of the endodermis by osmosis. The contents of the endodermis cells become more dilute. Water then moves into the cells of the pericycle. The content of the cells of the pericycle become more dilute, and water molecules move into the xylem vessels. Water is then conducted up the plant by osmosis and by various forces. Now these forces will be covered later on. On their part, mineral salts are dissolved in water found in the soil, and they are absorbed by active transport, a process that requires the usage of energy. However, a little amount of dissolved mineral salts may also be absorbed along with water through the root hair cells due to transpiration or pull, a force that we shall come to look at again. Mineral salts are more concentrated in the roots of a plant than in the soil, and they cannot be absorbed by osmosis, and hence mineral salts are absorbed actively by using energy. There are several factors that would affect the absorption of mineral salts, and we are going to look at them next. Any factor that affects the rate of metabolism will affect the rate at which mineral salts are absorbed in the soil. Why? Because the absorption of mineral salts is an active process that consumes energy, and metabolism is the process by which energy is liberated. Three factors will affect the rate at which metabolism occurs and subsequently the rate of absorption of mineral salts. Metabolic inhibitors. Metabolic inhibitors are chemical substances that prevent metabolic activities from taking place. These inhibitors therefore prevent the release of energy, and thus active transport does not take place. Examples of such inhibitors include poisons such as cyanide. Temperature. Metabolic activities are influenced by enzymes which speed up the rate of chemical reactions. Enzymes are temperature dependent, and at low temperatures, the rate of enzyme activity is low, and therefore the rate of metabolism is subsequently low. Increase in temperature up to the optimum speeds up the rate of chemical reactions. However, at extremely high temperatures, the enzymes are denatured, and therefore the rate of metabolism again is affected negatively. Oxygen concentration. Oxygen is necessary in active transport. Why? Because oxygen is used in oxidation of substances that yield energy for use in active uptake of mineral salts. Remember, active transport consumes energy obtained through metabolism. Metabolism involves oxidation of substances, and therefore the amount of oxygen is a factor in determining the rate of metabolism and subsequently the rate of absorption of mineral salts. Before we look at the next part of the plant, that is the stem, let's look at the vascular bundles and the structures. And we start with the xylem vessels. The xylem vessels are long hollow tubes made up of dead cells placed end to end. Their walls are thickened with lignin to prevent them from collapsing as water is transported up the plant. The thickening occurs in a variety of patterns. Some of the patterns are spiral, annular, and reticular. The hollow part is known as the lumen and provides passage for substances. The xylem walls have perforations which form simple pits. The lateral movement of substances is made possible through the pits, and the vertical movement is made possible by the lumen. As the vessels grow and develop, more lignin is deposited. The protoplasm of the vessels die and the cross walls disintegrate, and this results in the xylem vessel becoming long hollow tubular vessel. We also have elements we call tracheids elements. Tracheids elements are elongated with tapering ends. They are made of dead cells, and the tapering end walls of neighboring cells interlock. The tracheids elements are laid end to end, and this arrangement enables a stream of water to be continuous. The end walls of the tracheids have perforations through which vertical movement of substances occur. However, the movement of substances through the tracheids is less efficient compared to that of the xylem vessels. Note that the perforations would always slow down the movement of substances in the tracheids. The thickening of the tracheid walls with lignin and the interlocking tapering end walls provide mechanical support in the plant. The arrangement of the thickened xylem walls within the stem and roots form columns of reinforcing mechanical supportive tissue. The interlocking end walls allow the stem to bend without breaking. Parenchyma cells are also found between the xylem vessels and the tracheids. How about the phloem? Phloem transports carbohydrates and amino acids that are made in the leaf to other parts of the conduction is carried out by two types of elongated cells, sieve cells and sieve tube members. Both have clusters of spores known as sieve areas found on the overlapping ends of cell. These structures allow movement of materials from the leaves to storage parts, a process called translocation. Turgor pressure increases in the sieve tube members as sucrose from surrounding cells are brought into the phloem through active transport. Water then enters the phloem from the xylem by osmosis, which drives the transport of carbohydrates in the phloem. Movement in the xylem is one way, while in phloem, materials can go either way. Next is the stem. Substances in the stem are transported within the vascular tissue. The tissue are continuous with those of the roots, and the vascular tissue comprise the xylem and phloem. The arrangement of the vascular tissue in monocotyledonous and dicotyledonous stems differ. In a monocotyledonous plant, the vascular bundles in the stem are arranged randomly. Mature vascular bundles do not have a cambium. In a dicot stem, the xylem and phloem make up the vascular bundles, which are arranged around a central pith. The xylem are located on the inside while the phloem are located on the outside with the cambium in between. Next is the leaf. The leaf is the site of food synthesis but also plays a role in the transport of water. The arrangement of tissues in the leaf is designed to facilitate the movement of gases and water into and out of the leaf. A leaf contains several layers of specialized cells. At the very upper surface is an uncellular waxy coat called the cuticle. The cuticle offers protection against invasion by microbes and also serves to reduce the rate of water loss if need be. Next is the upper epidermis. The upper epidermis is 1 cell thick and provides a protective covering. Below the upper epidermis is the mesophyll tissue. Cells in the palisade mesophyll layer are sites for photosynthesis. They contain numerous chloroplasts. They are also sites for secretion and storage of food and water. The spongy mesophyll layer contains loosely packed cells, where spaces between cells allow gas exchange and passage of water vapor. On the lower epidermal tissue are the stomata, which are microscopic openings flanked by guard cells. Through the stomata, gases pass into and out of the leaf, as well as water vapor evaporating from the leaf, a process called transpiration. The spongy mesophyll layer contains arrangements of vascular tissues of xylem and phloem continuous from the roots, so water passes all through from the roots after absorption into the leaves, where it is lost through transpiration. Transpiration. Transpiration is the loss of water vapor mainly through the stomata in the leaves from the plant, but loss of water can also occur in liquid form. Loss of droplets of water from the plant is called guttation. Guttation occurs through special glands found mostly at the points where the vein is in contact with the edge of the leaf. The glands are called hydathodes. They are located on the leaf margin or apex. Guttation usually occurs in plants that grow in wet habitats. In transpiration, water gets to the leaves through the xylem vessels. The water leaves the xylem vessels and enters the cells of the spongy mesophyll by osmosis. This water then moves from cell to cell by the same process of osmosis and then diffuses into the substomatal airspace in the form of vapor. The concentration of water molecules is higher in the air spaces than in the atmosphere, and water diffuses out through the stomata into the atmosphere. Transpiration also occurs through the cuticle and lenticels. Lenticels are spores on the stems of woody plants that allow gaseous exchange between the atmosphere and the internal tissues. Stomata are found on both or either the upper or lower leaf surfaces. The distribution of stomata depends on the habitat of the plant. Plants that grow in hot and dry areas have most of their stomata on the lower part of the leaf. Those that grow in wet areas have stomata distributed evenly on both the upper and the lower part, but may even have majority of the stomata on the upper surface. Water vapor diffuses out of the leaf through the stomata. stomatotranspiration accounts for 90% of all the water loss from the plant. In cuticular transpiration, water vapor is lost directly through the waxy-like layer of cutin covering the surface of the leaves. A thick cuticle reduces the amount of water loss. Cutin is secreted by cells of the epidermis and makes up the cuticle layer. Factors affecting the rate of transpiration. Now, the rate of transpiration is affected by features on the plant such as the cuticle. Environmental factors also affect the rate of transpiration. Now, we start with features of the plant, and the first one is the roots. Plants with an extensive root system have a higher rate of transpiration than those with few roots. Why? This is because the extensive roots absorb more water, and therefore more is available in the substomatal space and therefore is lost through the transpiration process. Size of the leaves. More water is lost from the plants with large broad leaves than those with small narrow leaves. Why? Because those with broad leaves have huge number of stomata, and therefore the surface area through which water is lost is also high. An example of a narrow leaf is the pinus, and that of a broad leaf is the hibiscus. The large leaves have a large surface area over which transpiration takes place, and also the number of stomata is also higher. Leaf structure. The leaves have a variety of structures that affect transpiration. Now, these structures that affect transpiration include the following. First is the cuticle, and we've said a thick cuticle increases the distance through which water vapor diffuses, and hence less water is lost from such a leaf. The thinner the cuticle, the higher the rate of cuticular transpiration. The cuticle in most cases is waxy. Leaves covered with wax reflect light and hence low temperature in the leaf. Wax is also waterproof, therefore the presence of wax on a leaf epidermis reduces the rate of transpiration. Next is number of stomata, and the fewer the number of stomata, the lower the rate of transpiration. The more the number of stomata, the higher the rate of transpiration. Position of the stomata is also an important factor. Why? Because the sun shines directly on the upper surface of the leaves. The sun's rays increase the rate of vaporization from the upper surface of the leaves. Stomata on the lower surface are sheltered from the sun's rays, and thus more water is lost from the upper epidermis than the lower epidermis. Next, sunken stomata. When stomata are sunken, water vapor accumulates in the substomatal spaces. The water vapor is not exposed to moving air, and thus the rate of transpiration is also reduced and the leaf loses less water. Size of substomatal spaces and large air spaces between cells of the spongy mesophyll and stomata, called the substomatal air spaces, increase the rate of transpiration. Small substomatal air space reduce the rate of transpiration. Finally is the epidermal hairs. Water vapor on the leaves is trapped by epidermal hairs, and therefore less water vapor is lost from the leaf with more epidermal hairs through transpiration than a leaf with less epidermal hairs. The next category of factors is the environmental factors, and the first one is light. Light is instrumental in photosynthesis. Photosynthesis occurs in the presence of light, and a higher light intensity increases the rate of photosynthesis in the guard cells. Now, this results in the opening of stomata and hence a higher rate of transpiration, leading to loss of more water to the surrounding epidermal cells. Higher light intensity also increases the internal temperature of a leaf, and hence increasing the evaporation rate in the intercellular spaces, leading to a higher rate of transpiration. A lower light intensity will reduce the rate of transpiration. Next environmental factor is humidity of the atmosphere. When it is humid, water vapor in the atmosphere is very high than that in the intercellular spaces, and hence the water vapor diffusion gradient is low. Now, this means that the rate of transpiration is low when the humidity is high. When the atmosphere is dry, the water vapor diffusion gradient is steep and the rate of transpiration is high. The next environmental factor is temperature, and a rise in temperature will increase the water vapor holding capacity of air in the air spaces within the mesophyll cells. What this does is that it results in more water diffusing from the mesophyll cells to the air spaces, and thus water vapor pressure in the intercellular spaces increase. This causes an increase in diffusion gradient between the intercellular spaces and the atmosphere, and thus an increase in temperature increases the rate of transpiration. A decrease in temperature decreases the water vapor pressure in the intercellular spaces, and the diffusion gradient between the intercellular spaces and the atmosphere is lowered, and thus the rate of transpiration goes down. Wind is another environmental factor. When wind blows over the leaf, it carries the moisture away from the leaf, and this results in a steep diffusion gradient between the intercellular spaces and the atmosphere. Transpiration rate is therefore increased in windy conditions. When the air is still, the rate of transpiration is low. Next is availability of water in the soil, and water lost by plants should be replaced by water absorption through the root hairs. Plants wilt when water absorbed is less than the water lost through transpiration. There is adequate water in the soil, like during wet conditions, water is absorbed and conducted to all the cells. The mesophyll cells in the leaves become moist, and thus more water will diffuse into the intercellular spaces and increases the diffusion gradient. More water is lost to the atmosphere through transpiration. At the same time, the guard cells become turgid, forcing the stomata to remain open, and hence more water is lost through transpiration. When there is less water in the soil, the rate of transpiration is of course reduced. Finally is atmospheric pressure. At low atmospheric pressure, the rate at which water vapor diffuses between intercellular spaces and the atmosphere is high, and thus increases the rate of transpiration. At very high atmospheric pressure, the diffusion gradient between intercellular spaces and the atmosphere is low, and thus reducing the rate of transpiration. Plants that grow naturally at high altitudes where atmospheric pressure is low have modified leaves to reduce the rate of transpiration. Next, we look at the forces involved in the movement of water in the plants, right from the roots to the leaves. What forces drive that movement? Now, there are several forces that are responsible for the movement of water and minerals through the plant. Now, these forces include the following. The first one is transpirational pull, and transpirational pull allows or enables a stream of water to move from the roots up to the leaves. Energy from the sun causes evaporation of water from the plant through the stomata, the cuticle, or the lenticels. The diffusion gradient between the atmosphere and the mesophyll cells increases due to that, and water vapor diffuses into the atmosphere. The mesophyll cells then draw water from the xylem. The water in the xylem is replaced by a continuous column of water known as transpirational stream moving up from the roots. The other force is cohesion, and cohesion is the force of attraction between similar molecules. Water molecules are cohesive, that is, they stick to each other. Another force is adhesion, and adhesion is the force of attraction between different molecules. Water molecules adhere to the surface of the xylem vessel. Now, cohesion and adhesion forces maintain a continuous column of water in the xylem vessels from the root to the leaves of the plant. The next force is capillarity, and capillarity is the tendency of water to rise in very narrow tubes. The lumen of the xylem tracheids and vessels is very narrow, and this enables water to rise by capillarity. Capillarity is assisted by adhesion and cohesion forces as well. Finally is root pressure. Root pressure is the force that pushes water absorbed from the soil to move up the stem from the roots. The energy used to develop root pressure originates from the endodermal cells. Cells of the endodermis actively secrete mineral salts in the xylem. the osmotic pressure of the xylem content is increased. Water molecules move into the xylem through osmosis. So after all this, is transpiration really important? Yes, it is. Why? Transpiration is important because it develops a force or suction pressure that contributes in moving water up the plant. It also leads to absorption and distribution of water and mineral salts in the plant. Transpiration also causes the removal of excess water from the plant. It brings about cooling the plant when water vapor evaporates from the leaf. Finally, it causes wilting. Wilting is beneficial when a plant cannot obtain enough water to replace that lost by the plant through transpiration. Next, we look at translocation. Translocation is the movement of manufactured food substances from where they are manufactured in the leaves to the rest of the plant. Translocation occurs through the phloem tissue. The phloem is a vascular tissue made up of sieve tube cells and companion cells. Parenchyma cells fill up the space between the companion cells and the sieve tube element. The sieve tube cells is long with perforated end walls, which are called sieve plates or sieve areas. Cytoplasmic strands run through the sieve plates connecting adjacent cells. At maturity, sieve tube cells lack the nuclei and ribosomes. Mature sieve tube cells have fewer mitochondria. Some cytoplasm is also found in the periphery of the sieve tube cells. On the lateral walls are passages called the plasmodesmata. Substances move through the plasmodesmata from the companion cell to the sieve tube cells. A sieve tube cell is associated with one or more companion cells. Companion cells have thin cellulose cell walls. The protoplasm in companion cells has prominent nuclei, numerous mitochondria, rough endoplasmic reticulum, and Golgi bodies, and hence they have a dense cytoplasm. Translocation of manufactured food substances from the leaves to where they are either used or stored occurs in the phloem. Materials transported include carbohydrates such as glucose, fructose, maltose, and sucrose. Mineral elements such as nitrates, sulfates, phosphates, potassium ions are also translocated. Vitamins translocated include ascorbic acid, while hormones translocated include gibberellins and auxins as well. Phloem also translocates proteins and lipids. Manufactured food is translocated from the leaves to regions of growth such as root and shoot apices. Meristematic tissues, young leaves, flowers, and fruits require manufactured food substances for growth. Organic materials are also translocated from photosynthetic tissues such as leaves to storage organs and growing regions. Storage organs include the roots and stems. The roots and the stems may swell up with food to bulb and tubers. In an experiment to demonstrate translocation, a ring bark from a wood plant is cut off. The phloem tissue is contained in the bark while the xylem is in the woody part and remains intact. After a few days, a bulge of growth is observed on the upper edge and none on the lower end, and this is due to accumulation of food materials on transit.
High blood pressure is an indication of hypertension, and hypertension places people at higher risk for things like heart attacks, strokes, and kidney and heart failure, all potentially fatal conditions. Loss of pressure can mean insufficient blood flow and a lack of oxygen to the tissues, which can cause permanent damage, even death. It's very important to restore blood volume and blood pressure as soon as possible to provide oxygen to these tissues.